Welcome to another episode of Is Who of Enzymes. For today, we deepen our knowledge on a topic that we already spent a whole episode on, the cytochrome P450s. In detail, we will talk a bit about the non-natural activities that were investigated in the past, let's say roughly 20 years. I subtitled my talk based on literature quotes as a new job for an old enzyme. Also depicted here is a figure from the Nobel lecture of Frances Arnold, which nicely presents today's topic. We have a native enzyme that does an excellent job on its native reaction. Offering the enzyme a new job might cause a dramatic loss in performance, but still might already give a hint that in principle the enzyme is capable of doing this new job. Going through generations of enzyme evolution, the enzyme is then optimized for its new job. Before we start right into the topic, in case you haven't checked out the first episode on the natural reactions of P450s, here's a link, and also you can find it in the ElkTalk channel. Taking a look at the development of P450 research, there is a clear increase in the number of papers published every year, reaching more than 5,000 publications in the past year. If we consider the occurrence of P450 enzymes in nature, this is not surprising. P450s are found in every part of the tree of life, including humans, bacteria, viruses, algae, and plants in general, animals, including birds and spiders, and also fungi. And hand in hand with the strong presence of P450s in nature also goes a broad reaction scope that is found and considered as a natural reaction, including aryl-aryl couplings, ring contractions and expansions, dealkylations, decarboxylations, and so on. But what actually is a natural or a non-natural reaction, and how to distinguish between those? On the slide, you see a clear straight black line between natural and non-natural. But maybe this line should more be grayish, or even a dashed line. Or I would prefer a color gradient between natural and non-natural. Some reactions can clearly be identified as natural reactions. These are reactions that occur like they are in nature. And many reactions in the metabolism, including metabolism of drugs, xenobiotics, or eicosanoids, as well as steroid synthesis and metabolism or vitamin D3 synthesis, all of these processes somehow at some point involve a P450 enzyme. Some reactions also might clearly be identified as a non-natural reaction, for example, because they are based on a reagent that is either not found in nature or that would not be stable in nature. These reactions, for example, are acyridination, cyclopropanation, or also sulfimidation. And then there is this gray part in the middle. There are other promiscuities like substrate and product promiscuity and also enzyme promiscuity, where also the ELK crew published a paper on a peroxygenase showing desaturase activity. As all of this is quite a broad spectrum for today's talk, this would be out of scope, and therefore I decided to build today's presentation on 3 quotes out of literature. On one hand, I consider a reaction as non-natural if it is catalyzed by an engineered enzyme that has no natural counterparts, meaning a reaction that would not take place in nature, for instance, because the reagent would not be available or stable in nature. This is also what the second quote mentions, in addition to new reactive intermediates. And furthermore, if a reaction is known to synthetic chemists, but is not yet found in biology. This is the kind of frame that I apply on today's topic. In nature, the P450 enzymes use reactive oxygen intermediates, and the scope in general is based on biological reagent and natural selection. For the non-natural part, that is based on synthetic reagent and new intermediates like carbines and nitrines, as well as directed evolution. We can divide the up to now found non-natural reactions to abiological carbene transfer and abiological nitrine transfer. A carbene transfer enables the enzyme to perform inter- and intramolecular cyclopropanation, bicyclobutanation, cyclopropenation, and XH insertions. Furthermore, ChAmination, acyridination, and amino hydroxylation, as well as sulfimidation. But first, let's take a look at how non-natural reactions in general can be accessed. Between the reactions taking part in nature and the desires of a synthetic chemist is a large gap. Reaction that synthetic chemists are striving for would in many cases not take place in nature, either because products are not useful to living systems or the required reagents do not stably exist in nature, or because the conditions that are needed to affect the reactions are not available. To bridge the gap between the repertoire in nature and human desires, several attempts have been made to design new enzymes from scratch, based on knowledge of the reaction transition state structure. But given our limited understanding of the function of enzymes on the atomic level, alternative ways as engineering existing proteins had to be found. A way of unlocking new catalytic activities is to challenge enzymes with new environments or non-natural conditions, as well as synthetic reagents. Also by changing the enzyme itself, for instance by introducing a different metal center into the cofactor or by exchanging a few amino acids in the active site or close by, a completely new activity might be observed. These powerful molecular biology tools, such as directed evolution, can optimize and diversify biocatalyst function to the joy of a synthetic chemist. And also more and more rising in this field is machine learning. Here we see the structure of a wild-type P450 BM3 with the heme cofactor in the active site colored in red. And if the enzyme is now faded out, we see the heme cofactor even better. Why do I show the heme cofactor without the enzyme structure? Because it all started with metalloporphyrins. Metalloporphyrin complexes are already long used as synthetic models for cytochrome P450 enzymes, and they also catalyze chemical reactions that have no natural counterpart. For example, the reaction of an iron porphyrin and diazo compounds to create metal carbenoid reactive intermediates for cyclopropanation is known already for a long time and quite well documented. Also, the metal carbenoids could be isolated and gave evidence that the metalloporphyrin catalyzed cyclopropanation reactions proceed via the metal carbenoid intermediate. Breslow and co-worker also showed the inter- and intramolecular nitrogen transfer with an iron porphyrin treated with iminoiodinanes. And the follow-up report showed that a rabbit liver cytochrome P450 could catalyze the exact same nitrogen transfer with very low turnovers, with below 3 turnovers. Given the similarity of carbene and nitrene and oxine in terms of their electronic configuration and electrophilicity, Arnold and co-workers hypothesized that P450s may exhibit promiscuous activity for carbene and nitrene transfer reactions. What they did was that they added some ethyl diazoacetate to styrene in presence of the P450 BM3, which led to trace amounts of the cyclopropane product. But not only the P450 could catalyze the cyclopropanation, even free hemide would catalyze the olefin cyclopropanation in water. As the P450 catalyzed cyclopropanation showed diaster selectivity different from that of free hemine and exhibited some enantiomeric induction, it is suggested that firstly the reaction is taking place in the enzyme's active site, and secondly that the active site geometry gives control over the stereochemical outcome of the reaction. And this was the starting point of engineering P450s for non-natural reactions, giving the old enzyme a new job. Now let's have a look on how they developed and improved cytochrome P450s for the application as a cyclopropanation catalyst from this starting point. The reactions were performed under argon with 30 millimolar substrate concentration here. Using free hemine as a catalyst already gave 15% yield with 73 total turnovers. Here we have preferred the trans configuration and almost no enantiomeric induction. In comparison to that, using a P450 BM3 wild-type enzyme turned out with only 1% yield and only 5 total turnovers, but the difference is trans ratio and especially in the cis configuration, some enantiomeric induction, meaning more cis product, and out of the cis, the SR was preferred over the RS. Introducing one single mutation to the at position 268 resulted in a nice increase in the yield to 65% and more than 300 total turnovers, with a way better cis-trans ratio and also a nice enantiomeric induction here. If we in general take a look at the best outcome of each column, we see that this variant already shows really nice results. The last variant down here shows the highest enantiomeric excess for the cis configuration, and the variant above with A328G is the only variant in this table that shows the other enantiomeric excess. meaning the RS preferred over the SR. With this P450 cis variant and one additional mutation at position 438, as already mentioned, a nice cis-trans ratio of 92 to 8 and 97% EE was achieved. By exchanging the iron ligating amino acid at position 400 from a cysteine to a serine, the characteristic peak at 450 nanometers is shifted to 411, which is why these enzymes are then called the P411. With this variant, even a cis-trans ratio of 90 to 10 with 99% EE was achieved. Here we have a summary of the most important mutations. Changing the threonine at position 268 to an alanine improved the catalyst for cyclopropanation more than 60-fold. And in the native monooxygenation catalytic cycle, this threonine here is a key residue. Furthermore, we have this cis variant with one additional mutation, which led to further tailoring of the active site and which then exhibited excellent diastereoselectivity and enantioselectivity selectivity. And finally, as also already mentioned, the C400S mutation causes the shift of the 400 nanometer peak to 411, which gives name to the P411 enzymes. And in total, we can say that the very final outcome was a styrene cyclopropanation on a gram scale with 67,000 turnovers and 72% yield. Here we see the structure of P450 BM3 cis, and which we have seen is able to catalyze a cyclopropanation with 40% yield and 199 total turnovers. We nicely see the heme cofactor here in the active site with its iron, and zooming in you can see that I marked two residues. I marked position 400 in green up here, which is a cysteine. By mutating this amino acid to a serine, the characteristic peak is shifted from 450 to 411 nanometers, and marked in yellow down here is position 438, which by mutating the threonine to a serine resulted in further tailoring of the active site and gave excellent diastereo and enantioselectivity. Until now, we only discussed the cyclopropanation of styrene, but literature shows that this reaction is not limited to styrene as a substrate. Cyclopropanation was performed on acrylamides with variation on the amide moiety as well as on the aryl ring. For the amide moiety, there were variations in the range of methyl groups, methoxy groups, and different heteroatom ring systems. For the first 5 compounds given here, the yield is in all cases above 90% with total turnover numbers above 4,700 and nice diastereoselectivity and enantioselectivity. For the other 2 compounds down here, yield and also total turnover number was lower, but still the reactions worked pretty nicely. For the aryl rings, several variations were tested, including meta and para substitution, para-methoxy substitution, chloro compounds, trifluorocompounds, as well as this naphthalene derivative. And in total, for all of them, the yields were above 75% and 3,800 to 4,200 total turnovers with good selectivity. Coming to the last point of our cyclopropanation story, it was shown that the cytochrome P450 could be applied as a cyclopropanation catalyst in the synthesis of levomilinazepran, which is prescribed as an antidepressant and currently marked as Fetsima brand name. Now we come back to our little overview on the non-natural reactions, and until now we only had a look into the cyclopropanation reaction, which is the reaction the non-natural P450 reaction started with. But for the abiological carbon transfer, there are still a few more reactions known, namely the bicyclobutanation, cyclopropanation, and the insertion reactions. The bicyclobutanation is really nice as small carbocycles are structurally rigid and possess high intrinsic energy due to their ring strain. And these features enable small carbocycles for broad applications, but also result in challenges for their construction. For the bicyclobutanation, we start from phenylacetylene and perform 2 sequential carbon transfer reactions, which brings up quite a few challenges. First, the enzyme would need to bind the alkyne in a specific conformation in order to transfer the carbene enantioselectively. Second, the high-energy cyclopropene intermediate generated after or by the first carbene addition would need to be accepted and stabilized by the protein, and a substituted carbene might hinder access of the cyclopropene to the iron carbenoid. Testings of free heme gave no product formation. Further investigation of several P450s and P411 variants, cytochrome Cs and globins were also not fruitful. But a P411 resulted in the furane product that is also shown up here with a total turnover number of 210. And more detailed investigation of how the furan was achieved revealed that the enzyme most likely first synthesized the cyclopropene, which then rearranged the furan spontaneously or with the help of the enzyme. And that showed that the enzyme in principle is capable of transferring a carbene to an alkyne. And further screenings of P411 variants enabled them to prepare bicyclobutanes via 2 times carbene transfer. If we take a look at the mechanistic figure here, we start from the phenylacetylene, we perform 1 carbene transfer, we end up with the cyclopropene, which then either rearranges to the furane product, or if another carbene transfer is happening, we end up with the desired bicyclobutane product. Several substrates were also screened with decent yields and total turnover numbers, and a few more substrates were applied even in a millimole scale, with some more functional groups up here, which opens up possibilities for further chemistry. Also, the cyclopropenes possess high strain energy, which enables activity in different transformations for constructing several molecular scaffolds. Carbene transfer is one of the most straightforward approaches to constructing cyclopropenes. Small molecule transition metal complexes based on rhodium, cobalt, and others have been shown to catalyze carbene transfer to terminal alkynes. However, enantioselective carbene transfer to internal alkynes is largely unexplored, and only 2 systems with precious gold, silver, or rhodium catalysts have been reported for asymmetric cyclopropene synthesis. In the initial screening, they found 55 total turnovers with only a single enantiomer as product, and going through several generations of evolution, the P411 could be optimized for the cyclopropenation. Also here we find a substrate scope of mainly modifications on the aromatic ring. There is only 1 substrate which has 1 carbon less on the other side of the propene. And all of them turned out to be highly enantioselective. And furthermore, 2 more substrates were screened on a preparative scale of 1 millimole. Now we approach the last type of reaction for abiological carbene transfer, which is the HX insertion reaction. Several types of insertion reactions were investigated, some were optimized, and for some other heme enzymes like myoglobins or cytochrome Cs were more suitable. For the first reaction, we have an insertion into an NH bond with decent yields and 291 total turnovers. The second reaction is sulfur hydrogen insertion, which also achieved nice EEs and quite high total turnover numbers. The next 2 insertion reactions, silicon hydrogen and boron hydrogen, were both found to somehow occur, but were not further investigated as other heme proteins performed superior, especially for the boron hydrogen insertion. That one was found in traces only. And the last reaction is an insertion into carbon-hydrogen bond, which was found to proceed in a highly enantioselective manner with a high total turnover number. What we are still missing now are the nitrogen transfer reactions. Part of those are the ChAmination, acyridination, aminohydroxylation, and sulfimidation. The ChAminations were already reported more than 30 years ago for a mammalian cytochrome P450 preparation reacting with iminoiodinanes, although only on a trace level and with 3 total turnovers. As iminoiodinanes are difficult for biocatalytic applications due to their insolubility in aqueous media, sulfonyl acids were tested as alternatives. And as side product, and in the beginning of the screening, in many cases, the major product were the sulfonamides. Still, all of the tested enzymes, including the wild type, gave small amounts of ChAmination product, and finally, with a variant of P411, 430 total turnovers and 87% enantiomeric excess were achieved. The intermolecular benzylic ChAmination worked even better using tosyl azide as nitrogen precursor and accumulating beneficial mutations. Here, a P450 variant was optimized for this reaction, exhibiting up to 1,300 turnovers with up to 99% EE. Yeah. Screening several heme proteins for amino hydroxylation showed that indeed a P450 variant was able to catalyze the reaction, but gave low enantiomeric induction and only up to 4.4% yield. And as also in this case, a cytochrome C performed superior, the P450 variants capable of performing amino hydroxylation were not further optimized or evolved. The last reaction of abiological nitrogen transfer is the sulfimidation, where a sulfide is transformed to the corresponding sulfimide by insertion of a nitrogen. And the reaction is again performed with a tosyl azide and a P450 variant with up to 320 total turnovers. With this, we reach the end of the non-natural P450 derived reactions that I present today, and you might have recognized that I did not go into detail for mechanistic aspects so far. For the sulfimidation, for example, there is a proposed mechanism that begins with the iron-free heme gaining an electron from NADPH via the flavin cofactor of the reductase domain. Addition of the azide substrate here results in the formation of a formal iron-4 nitronoid, which can either be reduced by a subsequent electron transfer to the left side or can be trapped by sulfide to form the sulfimide product. It is hypothesized that a second electron transfer, so the left pathway, followed by protonation of the nitronate to generate sulfonamide, restores the heme iron to its ferric state, and an additional reductant is then required to return to the catalytically active ferrous state here. For all the other reactions, I want to stay quite general when it comes to mechanistic aspects, as the time frame would not allow to go into detail for all of them. As I mentioned already somewhere in my presentation, the nitrine and carbene have a similarity to compound 1 in terms of their electronic configuration and also electrophilicity. And in the native catalysis of the monooxygenation, the iron-free compound, together with oxygen and NADPH, generates compound 1, which is the active species here, while in the non-natural catalysis, an iron-2 species reacts with the nitrine or carbene precursor to produce the carbenoid or nitrinoid reactive intermediate and nitrogen. With this, I'd like to close today's episode on non-natural P450 reactions. I hope I could give you an overview which non-natural P450 reactions have been investigated until now, and for more details on any of the reactions, you might check the references. Thank you for your attention.